Hello, my name is Beverley Greenwood-Van Meerveld, and I'm the immediate past president of the American Neurogastroenterology and Motility Society and the current chair of the ANMS Institute. I'd like to welcome you to the ANMS virtual symposium tonight entitled Early Life Stress, How Does It Impact GI Function in Later Life? So the goal of these basic science virtual symposia is to keep you up to date with the latest research in the field of neurogastroenterology and motility until the time comes when we can all meet again in person at our annual meeting. And that reminds me to tell you about the ANMS 2021 meeting. It's going to be held in Boston on August the 13th to August the 15th. So far, we've had a record number of abstracts submitted to the meeting, and we look forward to this exciting meeting in person in Boston. So please plan on attending if you can. So today I'll be serving as today's moderator. And without further ado, I'd like to start the session tonight. So we have 3 speakers. Each will give a 15-minute presentation. And following those 3 presentations, we'll have our Q&A. I'd like to remind you to put, if you do have a question, to put your question in the Q&A panel as opposed to the chat box. And any questions that we don't have time to address, we'll address them in doc matter. So I'd like to now introduce our first speaker, Dr. Lin Chang. She's Professor of Medicine and Vice Chief of the Division of Digestive Diseases and serves as the co-director of the G. Oppenheimer Center for Neurobiology of Stress and Resilience at the David Geffen School of Medicine at UCLA. Dr. Chang previously served as the president of the ANMS and is currently on the advisory panel of the ANMS Institute. And the title of her talk today is Early Life Abuse and Other Adverse Life Events Impact IBS Symptoms in Adults. Thank you, Lynn.
Thank you, Beverly. I just really want to thank the organizers for inviting me to speak today on this topic. Association between early life events and symptoms of IBS in adults. The objectives of my talk are to review the prevalence of early adverse life events, or what I will abbreviate as EAL, in patients with IBS compared to healthy individuals. Also to describe the early adverse life event related factors that can increase as well as decrease the risk of having IBS. We're also going to elucidate whether EALs are associated with symptoms in IBS patients. This is a proposed pathophysiologic model of IBS that I often use and think about when I see patients. On the left side of the screen, you see the risk factors or factors that can lead to the onset of IBS, specifically genetic or familial clustering factors that can increase the risk of getting IBS. There's also environmental factors like abuse history or early adverse life events. There can be acute gastroenteritis in those patients with post-infection IBS. In the blue box, you see the different pathophysiologic mechanisms that have been described in IBS patients that show alteration in brain-gut interactions. Now, once the patient has IBS, there are triggers. 2 of the main triggers of symptoms in IBS patients are food and stress. And that leads to the symptom presentation, and then there affects their outcomes and consultation behavior. So I have 3 questions that I'm going to review. The first question is, does a history of early adverse life events increase risk of having IBS? This is a summary of work done back at least 25 years ago by Doug Drossman and others, where they studied the prevalence of abuse in patients with IBS, either compared to healthy individuals or individuals with other GI conditions, specifically organic or quote organic GI disease. And what they found in studies is that GI symptoms can be associated with different types of abuse: physical, sexual, and verbal and emotional abuse. 31% of patients with IBS reported some form of abuse in this study by Doug Drossman. 44% of women that were GI patients had a history of sexual or physical abuse. You can see in the figure that in another study that 41% of women reported abuse versus 11% in men. And you could see the second study where it's the prevalence of sexual abuse, again, more common in women than in men. So patients with a history of sexual abuse were 2.8 times, almost 3 times more likely to have a functional bowel disorder. I want to summarize some other information about sexual and physical abuse in GI patients. In one study that Doug Drossman performed, he compared patients that came into GI clinic with a functional GI diagnosis like IBS, but other functional GI diagnoses. And then he compared the history of abuse or prevalence of abuse in patients with structural diagnosis like IBD. And he found that abuse was increased in both groups. What was different is that the functional GI patients had a greater frequency of severe abuse. And severe abuse is defined as rape or sexual penetration, and where physical abuse was considered or perceived as life-threatening to that individual. So that was higher in patients with functional GI disorders versus patients with organic or structural GI disease. Now, independent of abuse history, functional GI diagnosis was associated with greater pain severity and greater psychological distress and poor daily function. Independent of the diagnosis, abuse history contributed to greater pain severity, more days in bed, more psychological distress, poor daily function. The type of diagnosis and abuse history independently contributed to the health status of the population. So when we see patients, we shouldn't just look at medical symptoms. We should also think of the whole other parts of their history that can impact their symptom severity and their global health status, like early adverse life events. When I started studying this area, because it is something that resonates in our patients, it's something you see in the clinic, I want to really measure other adverse life events and not just abuse. So there was a questionnaire that I first started off with, which is called the ETI-SR, and this is the Early Trauma Inventory Self-Report Short Form. It's been validated in PTSD, but it measures items of general trauma, physical abuse or punishment, emotional abuse, and sexual abuse. Now, this questionnaire just gives you a score of the number of items of each category that the patient endorses. It does not give you a categorization of whether if you meet criteria for a type of abuse or general trauma. And this questionnaire is used, and some people use early adverse life events before the age of 18 or up to the age of 18, or some studies have used under the age of 14. So they're really looking for events that occur before puberty. So we looked at this in our IBS patients and healthy controls, and these are basically individuals that were recruited from the community. So these aren't tertiary care patients. Separated the results between men and women. This is the data in women. Again, this is the prevalence of patients or healthy individuals that reported having at least one item. They endorse at least one item in each of these 4 categories. You can see it's pretty high. So women with IBS are shown in green, controls in blue. And in all 4 categories, there was a greater reporting of these different traumatic life events in women with IBS versus healthy controls. Now, when we looked at men, we had a lower number of men with IBS and healthy controls. And you could see that in this control group, this healthy men group, they had a higher prevalence of general trauma and physical abuse. And then emotional abuse is pretty comparable to women, and sexual abuse is lower in men than in women. So we didn't see a statistically significant difference, and it may have just been that the healthy men had endorsed more of these items, and also that we had a lower number of men than women. Now, Yuri Saito also used the ETI, and she studied IBS patients, their relatives, and controls. And she found pretty comparable results to us. Almost 75% of IBS patients experience any trauma, general trauma, compared to 59% of controls with an odds ratio of 1.56. When she looked at emotional abuse, 55% of IBS had emotional abuse versus 33.7% of controls. Sexual trauma and physical punishment were not higher in IBS versus controls in her study. What she also looked at was when the patients experienced trauma. Of course, this is retrospective, that they're reporting these items compared to when they actually develop IBS symptoms. The median onset of IBS symptoms were at age 25, and IBS cases were more likely to experience any general trauma prior to their IBS diagnosis, and a higher overall number of trauma items compared to controls. So the traumatic events preceded IBS diagnosis. I will say that I forgot to mention that in the abuse studies that Drossman and other people conducted, they looked at abuse in adulthood and in childhood. So it wasn't specifically early adverse life events. The questionnaire that's probably used the most now is the adverse childhood experiences, and we studied this as well. This is the questionnaire that is used in large scale studies, and it started in the Kaiser population in the United States. This is the questionnaire that most investigators are using now, like in depression. Also, they have a similar questionnaire that you can use in children. So what we found when we looked at it in our IBS patients and in healthy individuals is that they did have a high, and this also categorizes whether the individual meets the criteria for abuse or another adverse childhood experience. And we found that abuse was increased in IBS patients versus controls, and also a adverse childhood experience related to something that happened in the household. So that there was mental illness in the household they lived at. It's not that they just knew somebody, it was actually living with, that person was living in their household, and an incarcerated relative living in their household. If you had an adverse childhood experience, it increased your risk of having IBS by twofold. Now, this ACE questionnaire has been used in many chronic illness studies, and it does increase the risk of chronic illness and also negative health behaviors. And it's about twofold as well. It also has a graded relationship. The more adverse childhood experiences the patient has, the more likely that they're at risk for these chronic illnesses, including IBS. So you can see in the IBS patients, they had more ACEs or adverse childhood experiences than you see in healthy controls. The other question is, what early adverse life event related factors modify the risk of IBS? We know that many individuals have an early adverse life event. They don't all get IBS. They don't all get diabetes or cardiovascular disease or depression. So what makes someone more at risk? So we looked at it by a questionnaire called the CTES. It not only asks about traumatic events, it asks about the severity of that traumatic event. And we calculated something called the total trauma burden. So what that is it's the sum of the trauma severity ratings for each traumatic event and the number of events, and found that total trauma burden increase the odds of having IBS than just experiencing trauma. So the severity of that traumatic event was very important. Now, if you look at the odds ratio, it's only about 11% increase, but it did increase the risk of having IBS. The other aspect that's important about a traumatic event in predicting IBS status is whether fear was associated or perceived by that individual related to that traumatic event. If you see this left panel, 60% of IBS patients reported that an early life traumatic event, this is using the ETI, the first questionnaire I showed you, they reported having fear associated with that traumatic event, while 36% of healthy controls reported fear. This is also whether they had, this is the difference between men and women, which you see no difference in. Now, if someone reported having fear associated with a traumatic event, that increased the odds of having IBS twofold. So it further increased just the risk of having a traumatic event, but it further increased the risk of having IBS if they had fear associated. So it started this whole fear response pattern and this helplessness that we use in animal models. Now, we don't study many protective factors related to early adverse life events. In this study, we found that when an individual reported confiding in others about that traumatic event around the time that it actually occurred, they had a decreased odds of having IBS. And this is really important because it tells us that if something happens to an individual, can we intervene to decrease the odds that they'll get IBS or another chronic illness with, for example, like behavioral therapy. Are early adverse life events associated with symptoms in IBS? And I try to summarize the different abuse or adverse childhood experience questionnaires or interviews and summarize the data. So in the abuse history studies, they found that abuse history did increase pain severity, days in bed, psychological distress, and reduced daily functioning. You can see the ACE questionnaire that was associated with increased symptom severity, abdominal pain, and also somatic symptoms. The childhood traumatic event scale also showed a strong trend to being associated with IBS symptom severity and abdominal pain. The ETI didn't really correlate with symptom severity of IBS, but having fear related to the traumatic events was associated with more anxiety and lower mental health related quality of life. So to summarize, IBS patients report stress as a cause and as a trigger of their symptoms. Early adverse life events increase the risk of having IBS, and the ones that have been associated with IBS are sexual and emotional abuse. And I will say in studies, emotional abuse is one of the strongest predictors of getting IBS. Mental illness in the household, incarcerated family member increases risk. Increasing number and the severity of the early life event and associated fear further increase the risk of IBS on top of just experiencing that traumatic event. Confiding in others about the trauma decreases the risk. Early adverse life events may be associated with increased GI symptoms, including pain, worse health status, and psychological distress. So recognizing early adverse life events in our patients is important to guide management and improve health outcomes. So thank you very much for your attention, and I'll be happy to answer any questions during the Q&A.
Thank you. Thank you, Dr. Chang. Our next presenter is Dr. Tijs Louwies. Dr. Louwies obtained his PhD in 2016 from Hasselt University in Belgium. And in 2018, Dr. Louwies joined my lab at the University of Oklahoma Health Sciences Center as a postdoctoral fellow. And the title of his presentation today is Early Life Stress and Sex Differences in Brain Epigenetic Regulation of Chronic Visceral Pain. Thank you, Dr. Louwies.
Well, Dr. Greenwood, thank you for the nice introduction. I would also like to thank the organizers for this opportunity to present my research on how early life stress can cause chronic visceral pain later in life. As we have learned during Dr. Chang's presentation, early life adversity can increase the likelihood of developing functional gastrointestinal disorders later in life, such as irritable bowel syndrome. Now, we know from epidemiological studies that many IBS patients do report a history of early life stress, whether it is growing up in poverty, being abused, or experiencing trauma. Now, interestingly, as Dr. Chang has pointed out, the rate of childhood abuse is much higher, up to 2 to 4 times higher in IBS patients than in healthy controls. And has also been shown in Dr. Chang's presentation, females may be more susceptible to the negative effects of early life stress. And this may suggest that there is an important role for the gonadal hormones. Now, if we specifically look at irritable bowel syndrome, unfortunately, we have to say that the etiology is still unclear. However, we do know that during colorectal distension, the amygdala of IBS patients becomes hyperactive when compared to normal controls. Now, this should not be surprising if you know that the amygdala, as part of the limbic pain centers, plays an important role in the processing of visceral nociceptive information. Now, other studies have shown that early life adversity also is associated with stress-related disorders later in life, such as PTSD, depression, and anxiety disorders. Now, this association is mediated by hyperactivity of the amygdala. In these specific brain imaging studies, they have shown that the number or the severity of early life adverse events correlates nicely with amygdala activity and the severity of the stress-related disorder later in life. However, up until this point, in human subjects, it is still unknown whether early life adversity can lead to hyperreactive amygdala, and which could be a cause for the development of irritable bowel syndrome. However, as we also know from different studies, depression and anxiety disorders are often comorbid with IBS, so therefore there might be a link. Now in our lab, we have shown that in preclinical models, early life stress can indeed lead to visceral pain in adulthood through a mechanism that involves the amygdala. And therefore, in the remainder of this presentation, I would like to take you through some of our exciting data that gives support for this association. Now, before we dive into the molecular mechanisms, I would like to present a few animal models of early life stress. We know that maternal separation has been used to mimic neglect, and the limited nesting material model can be used to mimic poverty. During maternal separation, the pups are separated from the dam for several hours a day for multiple consecutive days. And the limited nesting material model is based on the fact that all the nesting material is removed from the dam's cage and being replaced by a paper towel. Now, another model is the odor shock conditioning model, which indeed can mimic an abusive caregiver. In this specific model, the pups are exposed to a predictable shock or an unpredictable shock following odor exposure. Where maternal separation and limited nesting material can lead to behavioral changes in the dam, later in life they give rise to a male-specific visceral hypersensitivity phenotype. Only the odor shock conditioning model will give rise to a female-specific phenotype. In this model, after unpredictable neonatal early life stress, female rats develop visceral hypersensitivity when compared to the predictable early life stress controls and the odor-only controls, and also to all their male counterparts. In this way, by using this specific model, we are able to recapitulate the female predominance of irritable bowel syndrome. Now, in order to verify whether this model was truly female-specific, we performed ovarectomy on adult female rats that were previously exposed to unpredictable early life stress. And we observed that after removal of the ovaries, visceral hypersensitivity was completely gone. Now, when we administered estradiol to these overrectomized animals, we were able to reinstate visceral hypersensitivity, but only in the animals that were previously exposed to unpredictable early life stress. So this shows that visceral hypersensitivity in adult female rats after unpredictable early life stress depends on cycling estrogen. And in this way, we also have a model that we can use to study the role of gonadal hormones in the association between early life stress and outcomes in adulthood, and specifically look at how estrogen can explain this female or the female susceptibility for the effects of early life stress. Now that we have a working model in which early life stress is associated with visceral pain in adulthood, we wanted to investigate the role of the amygdala in this association. We specifically investigated the role of the central nucleus of the amygdala because this is the nociceptive amygdala. Also, this region contains a high density of neurons that express corticotrophin-releasing hormone, or CRH, which is known to be an important mediator of visceral hypersensitivity, both in the central nervous system as in the periphery. From the literature, we also know that glucocorticoid receptor, or GR, is an important negative regulator of CRH expression. So therefore, we wanted to investigate how early life stress can change the gene expression of these genes. Now, when we isolated the central nucleus of the amygdala of the male animals, we observed that there was no change in GR or CRH expression. This should not be surprising given the fact that there is no visceral phenotype to start with. However, when we looked at the amygdala of the female animals, we observed that both after predictable and unpredictable early life stress, GR expression was increased in the central nucleus of the amygdala of these adult females. However, only unpredictable early life stress did increase CRH expression, in the amygdala of these animals. Now, as I said, CRH is an important mediator of visceral hypersensitivity. So therefore, we wanted to investigate whether knocking down CRH expression in the central nucleus of the amygdala would be able to attenuate visceral hypersensitivity. As you can see, by using a random sense inhibitor, which did not actually target CRH, we were not able to rescue visceral hypersensitivity. However, when we used the antisense inhibitor, which paired with a sequence of CRH and knocked it down, we were able to completely reverse early life stress-induced visceral hypersensitivity. So now that we know that early life stress can change gene expression later in life, the next question that we wanted to answer is, how can events that occur in the early life be transferred or carried over into adulthood? One potential explanation may be found in the epigenetic makeup of the amygdala. Epigenetics, which is the study of heritable changes in gene expression that do not involve changes to the underlying DNA sequence, or simply put, epigenetic mechanisms are responsible for making genes more or less accessible for gene transcription. These changes in the epigenome can change a phenotype without actually changing the genotype. Epigenetic mechanisms such as histone modifications, DNA methylation, and RNA interference are being used by cells to make genes more or less accessible for transcription. Now, the interesting thing is that these epigenetic mechanisms also allow cells to interact with the environment. By changing their epigenome, cells can change the gene expression when, for instance, the environment changes. Another interesting feature of epigenetic mechanisms is that they are often very persistent, and that changes in the epigenome can be maintained even in absence of the initial triggering event. So this can lead to long-lasting changes in gene expression. Now, in our lab, or over the past 2 years, I have been studying the role of histone modifications, and I specifically looked at the acetylation of histone 3-lysine 9. Histone acetylation is mediated by epigenetic enzymes such as histone acetyltransferases, which add acetyl groups to the lysine residues of the histone tails. Now, increases in histone acetylation are often associated with increases in gene expression. So, if we assume that early life stress is just an environmental signal, it can change histone 3-lysine 9 acetylation of certain genes. Now, this will lead to changes in gene expression, which can underlie, for instance, chronic visceral pain if this occurs in the central nucleus of the amygdala. So whether to test if histone 3-lysine 9 acetylation was actually involved in our visceral pain phenotype, we isolated the amygdala of these animals, both males and females, and we performed a Western blot for histone 3-lysine 9 acetylation. As you can see here, male rats do not show any changes in histone 3-lysine 9 acetylation. However, in adult female rats in the amygdala, we observed a significant increase after unpredictable and predictable early life stress. What I just showed you was global histone 3-lysine 9 acetylation. However, we also looked specifically at the histone acetylation status of the GR promoter, after we observed that after predictable and unpredictable early life stress, histone 3-lysine 9 acetylation was increased. Since histone 3-lysine 9 acetylation is a marker for increases in gene expression, these epigenetic changes corresponded with our observed changes in GR expression. Now, in the amygdala, at the CRH promoter, in contrast, we observed that GR binding was decreased after unpredictable early life stress. At the same time, histone 3-lysine 9 acetylation at the CRH promoter was increased. As I told you before, GR is an important negative regulator of CRH, so the loss in GR and the increase in histone 3-lysine 9 acetylation may have been the reason why we observed an increase in CRH expression in the amygdala of these animals. Although epigenetic mechanisms are often very persistent, that does not mean that they are not reversible. There are several small molecule inhibitors which we can use to target epigenetic enzymes, such as our histone acetyltransferases. So therefore, we wanted to test whether inhibiting these histone acetyltransferases with carcinol could ameliorate early life stress-induced visceral hypersensitivity. So after administration of carcinol directly into the amygdala, we were able to successfully attenuate unpredictable early life stress-induced visceral hypersensitivity in these adult female rats. Now, on the molecular level, we observed that after carcinol administration, histone 3-lysine 9 acetylation at the CRH promoter was reduced, and also at the same time, GR binding at the CRH promoter was increased again. So taken together, the combination of a reduction in acetylation at the promoter and an increase in negative regulation may have led to the decrease in CRH in the amygdala and the attenuation of early life stress-induced visceral pain. Now, in order to summarize this talk, I would like to give you a take-home message. We know from epidemiological studies that early life adversity is associated with the development of irritable bowel syndrome later in life. Now, our preclinical data suggests that following exposure to early life adversity, epigenetic remodeling in the amygdala can contribute to chronic visceral pain in adulthood. And this may also be a mechanism that is happening in human patients. For the final seconds of this talk, I would like to thank my mentor and the moderator of this symposium, Dr. Greenwood-Van Meerveld, for her support during my postdoc, and the other members in the lab. And that being said, I would like to yield my time and give the word back to Dr. Greenwood.
Thank you, Dr. Louwies, for that excellent presentation. Before we start our final presentation, I'd like to remind people attending this session tonight to put any questions that you may have in the Q&A box. That would be nice. Thank you. So it's my pleasure now to introduce our final presenter of the evening, Dr. Siobhain M. She's currently a senior lecturer in the Department of Anatomy and Neuroscience at the University of Cork in the Republic of Ireland. And the title of her presentation today is Nutritional Interventions During Pregnancy to Reduce the Impact of Stress on the Microbiome-Gut-Brain Axis. Thank you, Dr. Mahoney, and thank you for joining us this evening because we know it's very late for you in Ireland.
Thank you very much, Beverley, for a lovely introduction. So I also want to thank the organisers as well for the invitation to speak today. Okay. So as Beverley stated, today I would like to speak about nutritional interventions during pregnancy in order to reduce the impact of stress, particularly related to its impact on the microbiome-gut-brain axis of the mother and then the developing baby. So this is just a very brief overview of the talk I'm going to give today, and I'm going to focus a tiny bit on the gut microbiome and how it changes during our lifespan. How it's important during pregnancy with regard to the mother and the child. We'll talk about the microbiome-gut-brain axis, and then of course stress and anxiety during pregnancy and interventions to reduce this. So as humans now, we're often viewed as holobionts, and holobionts really are the host and all of the microorganisms that live on and in it, basically, residing really as this kind of discrete ecological unit. So our gut really hosts, we have a number of different types of microorganisms on and within our body, but our gut really hosts the largest community of microorganisms of the entire body. So our gut microbiota, particularly the bacteria within our bowel, are associated with essential processes of our body, including metabolism, immunity, and well-being. And we now know there's about 1:1 with regard to ratio of bacterial cells in our body to human cells as well, whilst the genome of the microbiome is about 150-fold greater than that of the actual human genome. Now, our microbiome within our bowel changes during our lifespan. And as I mentioned, I just mentioned a few key processes our gut microbiome are involved in, and they're involved in many more than I've mentioned there. But during our lifespan, of course, there's different demands placed upon our body depending on that life stage and depending on what we are doing, depending on our hormones, depending on our activities, of course, as well. So our gut microbiome, just like us, changes during our lifespan. So the type of different bacteria changes. So we have different bacteria during life stages such as gestation, infancy, puberty, and so on, and again, old age. So it's not only the type of bacteria that change, but also the diversity of bacteria alters as well during our life stages. And that is basically that we have just reduced or increased numbers of the different types of bacteria. Now, our microbiome-gut-brain axis is a bidirectional signaling pathway between the brain and the gut. And it allows signals that are set up within the brain, such as psychological stress, to be perceived by the bowel and vice versa. So elements within our bowel or alterations or infections within our bowel that occur can be perceived by our brain and, of course, stress us out slightly. So the mechanisms that allow communication between these two end organs include our microbiota and all of the products they produce, including short-chain fatty acids here, for example. The other systems that allow this communication to occur include our immune system, our stress axis, which is our HPA axis, and our neuronal axis. Very importantly for our life, basically, and our health in adulthood, is the programming of this microbiome-gut-brain axis, because we know our microbiome is involved in essential processes. It's also involved in programming of the axis itself and allowing for the bidirectional signaling pathway to develop appropriately, and all of those mechanisms that I've just mentioned to develop appropriately as well. So different stages in a very early life, including prenatal period, and elements during our prenatal period, including diet, drugs, and infection, on the mother as well, including the mother's gut microbiome and vaginal microbiome, can impact on the developing baby. The largest time or the largest impact on the developing microbiome, and hence developing microbiome-gut-brain axis, is of course our mode of delivery. And we know that babies born vaginally or C-section have different microbiota profiles. Postnatal environmental factors also impact on our microbiome and developing microbiome-gut-brain axis. But it's not just the programming of this axis that's important. It's also can be influenced even if we have a perfectly functioning microbiome-gut-brain axis, which functions without you knowing if it's functioning perfectly. We also have influences in adulthood that can impact on this axis as well and lead to changes in any of the elements I mentioned. And these include stressors in the environment you live in, whether you live in an urban environment or a city or a country dwelling environment. Sex, diet, and health and disease all impact on our microbiome-gut-brain axis. Now, I mentioned that microbiome within our gut and changes during different life stages, but there's also different niches within the human body, including the oral microbiota or the oral cavity, the placenta, and the vaginal niches as well that change, their microbiome profile change during pregnancy. Now, the reason for our gut microbiome changing during pregnancy is to allow efficient energy harvesting from our food and also to dampen the immune response to allow the fetus to grow, of course. But we have changes to the vaginal microbiome as well during pregnancy in order to allow seeding of the baby's gut during birth. Now, we know that stress and anxiety during pregnancy lead to altered developmental outcomes in the developing baby, such as changes in cognition, anxiety, inflammation, and neuromotor function. Now, there has been proposition that these neurodevelopmental outcomes or altered neurodevelopmental outcomes may be due to the stress impacting on the microbiome of the mother. And as I mentioned, there's different niches obviously within any body, but also within the pregnant mother as well, including the oral cavity, the gut, and the vaginal cavity. And these are particularly impacted by stress, not only during pregnancy, but also during different life stages as well. But these stress during pregnancy may be particularly important with regard to changing the gut microbiome, which may influence the development of the developing baby. Now, we carried out a study a few years ago at this stage, but it was carried out basically on a larger cohort. And what we did was we drew down from individuals that had higher and lower levels of lower level scores on the Edinburgh postnatal depression scale. So these were a set of pregnant mothers, to be mothers, and we took samples, basically fecal samples, during the second and third trimester. And as I said, we categorize these moms to be into high and low scoring on these depression questionnaires. We also carried out a vaginal swab, but I won't present that tonight. We also carried out fecal sampling of the baby that were born to these mothers with the high and low stress, high and low anxiety, or high and low depression at 1, 2 weeks, 3 weeks, 3 months, and 5 months. I'm only presenting the diversity tonight with because of time, and another time I will catch you up on the whole study. So tonight, what I'll present tonight is the diversity during the 2nd and 3rd trimester from the fecal samples from the mothers. So in black, you see the mothers that had low scores, and the mothers in red, or the bars in red, are the mothers that had high scores, basically, for depression. So what we noticed was there was a decrease in diversity, basically, in the mothers that had high depression scores, but only during the 2nd trimester. Now, we know as adults, an increased diversity is associated with a more healthy individual. As I said, we didn't see this, interestingly, during the 3rd trimester. We did see an altered microbiome diversity in the infants born between whether they were born to mothers with high depression or low depression. But maybe the most interesting point, or one of the points that has been missed as well along the way, is that what we saw was changes to the mother's gut microbiome during the 2nd trimester, but not closer to birth. So all of these babies were born vaginally. So it wasn't necessarily the seeding at birth, which 3rd trimester microbiome would more or less indicate, but it was the stress and the microbiota diversity during the 2nd trimester that seemed to have the correlation with the baby's microbiome post-birth, indicating perhaps that the stress during pregnancy, altering the mother's gut microbiome, potentially vaginal microbiome as well, led to changes in the actual physiology of the bowel of the baby, which led to altered microbiome taking up residence, or influencing the microbiome that take up residence. Now, stress just doesn't just affect our microbiome, but it also affects different parts of our brain-gut axis as well. And one other element of our brain-gut axis it also impacts on is our barrier function, our epithelial barrier. So changes in a stressed microbiome-gut-brain axis to our barrier function leads to increased permeability, which leads to changes then in our systemic immune system, potentially because antigens or bacterial products or bacteria themselves leak out into the systemic immune system, increasing inflammatory responses in the systemic blood, which then, of course, we know can impact on the central nervous system, changing behavior and cognition and mood, of course. So we carried out a study during pregnancy in order to assess the impact of different types of depression, stress, and anxiety scores, basically, on a mother's gut permeability. So this was the same cohort, but different mothers than the previous study I've just mentioned. So again, plasma samples were collected at 15 weeks and 20 weeks of gestation. We had a total number of 104 individuals that we were able to categorize as having high or low perceived stress, high or low anxiety, or high or low depression as well. We also had a moderate category as well that we were able to categorize these individuals into. Now, interestingly as well, it wasn't all the same mothers to be that had high stress, high anxiety, and high depression. Different individuals had low or high anxiety, basically. So they don't all mean if you have high stress score, it doesn't mean you'll have a high anxiety or a high depressive score, at least during pregnancy and at least in our cohort. So this study was carried out. These samples were taken from individuals that initially signed up to the screening for pregnancy endpoints, and this study was carried out in Cork University Hospital. So we collected blood samples at 15 and 20 weeks gestation, and what we did was we analysed these blood samples then for indirect markers of gastrointestinal permeability. So what we noticed was that individuals, mothers that had higher levels of stress, anxiety, and depression, particularly depression, were associated with changes in lipopolysaccharide binding protein, SCD14, CRP, TNF-alpha, and IL-8, for example. So these are all indicators, indirect indicators of increased gut permeability, indicating that potentially depression, this is particularly the depression scores that were higher, indicating these changes here. So particularly depression scores are associated with changes in indirect markers of gut permeability. What this basically means is that we can have changes to inflammatory profile, systemically, changes in tryptophan systemically as well. All of these can potentially impact on the developing baby. Now, with regard to interventions for pregnancy, with regard to mood, anxiety, and depression, well, the antenatal period itself is a very poorly researched area, and often mental health, particularly in Ireland where I live, is really a taboo topic. So women, particularly during pregnancy or in the prenatal period, do not want to present with mental health problems. Now, non-pharmacological treatments are limited. Pharmacological interventions are often avoided, obviously because of potential heterogeneity. Now, of course, there are non-pharmacological interventions, alternative ones, such as cognitive behavioral therapy, but there have been a lot of mixed results, and there are very long waiting lists as well. Now, I'll just briefly mention this, but what I want to point out basically on this slide is that there have been some studies looking at maternal gut microbiome targeted interventions in pregnancy, but a lot of these really have focused on different scenarios, such as the reduction in gestational diabetes, changes in bowel habit, vaginal infections, for example. And there have been both preclinical and clinical studies looking at prebiotics and probiotics during pregnancy on these different outcomes, but not necessarily mood or stress. Now, very few studies have really focused on the impact of changing or modulating the gut microbiome during pregnancy to actually alleviate stress and reduce stress, anxiety, and depression, for example. These two studies were two of the rare studies that were carried out looking at the impact of different microbiota targeted interventions. So these types of interventions really are termed or coined as psychobiotics. And we know that psychobiotics now are beneficial bacteria, which are probiotics, or the support for these types of bacteria, which are prebiotics that influence bacteria-brain relationships. So as I said, very rarely there have been studies, but this probiotic Lactobacillus rhamnosus was shown to decrease levels of symptoms of depression, anxiety in the postpartum period when it was consumed during pregnancy. Also, prebiotics haven't been investigated with regard to mood during trying to reduce stress, anxiety, or depression during pregnancy. But what prebiotics do is they offer this very nice kind of alternative to probiotics during pregnancy. Because as I mentioned, during pregnancy, we already have this change in the mother's, potential mother's, gut microbiome and vaginal microbiome that are deliberately occurring to assist with pregnancy, to assist with the developing baby, and to assist with birth, of course. So taking a probiotic specifically tested in non-pregnant people, potentially in men, that has shown to reduce stress might not be suitable during pregnancy. While a scenario such as prebiotics, for example, is going to supplement or is going to enhance the bacteria that is already there, including bifidobacteria and lactobacillus, which we know are extremely important during pregnancy and also in the developing baby. So this is just to highlight that research aimed at delivering accessible, safe, effective treatments with the aim of improving psychological health during this extremely vulnerable time window is critical to improving maternal health and reducing psychological morbidity. So just as a take-home message or a take-home diagram, sometimes it's nice to present this visually as well as kind of with text. So during pregnancy, what we tend to see is we know that stress, anxiety, and depression during pregnancy basically change the mother's microbiome, change systemic inflammation, and we know it increases gut permeability. We've also shown that it changes tryptophan levels, and we've also shown that it changes, as I said, altered gut microbiome. It can also change programming of the fetus as well due to changing the placenta, available micro and macronutrients, and also changes or alterations or increased bacteria and amniotic fluid have been seen. Now, stresses during pregnancy obviously program the fetus, lead to behavioral deficits, neurodevelopmental outcomes, and change potentially the microbiome that is going to reside in the baby and take up colonization. These all have long-term impacts, as we know already. So there is a potential still. There have been very few studies looking at dietary interventions during pregnancy, but there is a potential, as we know now, with the background in the microbiome-gut-brain axis outside of pregnancy. Now the time is to actually investigate it during pregnancy to reduce stress, potentially with safe, effective management interventions, including dietary interventions such as prebiotics. With that, I want to thank you very much for your time, and thank you very much to the organisers.
Thank you, Dr. Mahoney. Can I ask each of the presenters now to turn on their microphones and their videos, please start their videos. And it's time for the Q&A, and may I ask I'll ask the first question that's come up. It's a question for Tijs. Do you have any thoughts on why there are sexually dimorphisms in the type of stress that affected visceral hypersensitivity? Have you looked into whether acetylation in the CEA leads to changes in sex hormones or the estrogen axis?
That is an excellent question. We have, if we look at the literature, we know that, for instance, the maternal separation model and the limited nesting model, they can lead to a premature activation of the HPA axis, whereas the odor shock conditioning model leaves that intact, or that the stress hyperresponsive period. So basically, in the other two models, you have an increase in corticosteroids or cortisol in these rats prematurely, which can mess up neurodevelopment. Whereas in our abusive model, we are only messing up the amygdala. Now, for the part of the influence of estrogen, we did look at estrogen levels in adulthood, and we did not see changes between the three groups. There was a lot of chatter in the data, which was probably due to the variation in due to the estrogen cycle. But we do know that in the CRH promoter, there are estrogen responsive elements. Whether they are active or not, that's still debated in the literature, but that might also be a role. That might also play an important role. So to answer I hope that answered your question. And if not, then I will gladly expand on this in the doc matter.
Thank you, Tijs. I don't see any other questions in the Q&A, so if people could put their questions in the box, but in the interim, I actually have a question that any of the panelists, or perhaps all of the panelists, could address. You know, early life stress is early life is such a critical time for development of the enteric nervous system, the central nervous system, the gut microbiome. And early life stress obviously could have effects at multiple levels. And my question to each of you is, where do you think our research efforts at this point? This is such a complicated topic. How should we focus our research efforts to delineate the underlying mechanisms by which early life stress has these prolonged changes that subsequently lead to poor health outcomes in adulthood? I think each of you will have a slightly different take on that question, but I'd really appreciate it. So perhaps who would like to start? Perhaps Lynn, you could take it away.
That's such a big question. You have to really start from the mother, pregnancy, and follow babies to children and teens, and along the way you really have to do a prospective study. And you probably have to look also at parents and children to see what they inherited and what has changed according to what their phenotype is. So my impression is that you have to look at different mechanisms. Like, for example, if you're just interested in histone acetylation, you would have to study that obviously more peripherally. You couldn't do it in the brain. But we're looking at DNA methylation, and we're looking in the colon and the immune cells. So taking some actually biologic measure and following it over time and seeing how it affects people when they develop symptoms or flares, and also comorbidity. So to me, it's really complex, but a longitudinal study is probably the way to go.
I agree. These longitudinal studies are absolutely essential. Tijs, what's your take on that? Where are you going next with your epigenetic modeling?
Well, I would like to add a few things on what Dr. Chang mentioned. Okay. We definitely need longitudinal studies. And with my background in animal models, I would argue maybe it's also a time to look at longitudinal effects. At this point, I have only published the data of what happens in adulthood. But these critical periods during right after early life stress, or in adolescence, when sex hormones come into play, that can also skew your epigenome towards a pro-nociceptive state or an anti-nociceptive state even. So therefore, I would support Lin's conclusion in saying, like, I would look at like different time points and also not limit myself to just the CNS, but also see how these changes in the CNS can correlate to peripheral changes in our animal models and whether those are transferable or translatable to like humans. So to have like a better comprehensive overview of what's going on.
Thank you. Siobhain, what's your take on this?
Yeah, no, it's a really, really interesting question, of course, and you know, it would be fantastic if we could get to that place. I suppose maybe, you know, one of the stages that hasn't been investigated in a huge extent is the preconception period, particularly in, you know, both males and females. And we know that stressors prenatally have some impact on the developing baby, but it hasn't been investigated with regard to its impact on the developing and long-term impact on the microbiome-gut-brain axis or the microbiome and enteric nervous system, for example, particularly with regard to both partners or both elements of contributing to a developing baby. So I think maybe the preconception period, which hasn't had much, I suppose, light up until, you know, a few years recently with regard to this, could be a good time to start investigating. But what it also means is that our longitudinal studies are going to get longer, which isn't making things more difficult. But, you know.
Along those lines, there is a question. Are the critical periods of development different for the ENS versus the CNS? And would this affect potential interventions to alleviate earlier life adverse events?
I can answer that if that's
okay. Sure.
Anybody else? Yes, they are different. They do overlap quite significantly, but they are different. So we know the developing enteric nervous system, developing gut and the developing enteric nervous system is mostly developed by birth. It can be modulated post-birth, of course, but with regard to trying to compare the central nervous system to the enteric nervous system, the CNS has a much more long life with regard to development. The central nervous system is actually still growing, so your brain is even still growing much into your puberty period, and also there's a huge synaptic pruning as well in the pubertal period as well with regard to the central nervous system. So there's a much more long-lived impact on the CNS, but I would still say that there is an interaction between the two, and we shouldn't see them as separate ever. And that one event in one system will impact on the other. And just because the enteric nervous system has developed much more, I suppose, matured much more before the central nervous system, doesn't mean still that psychological stressors can't really change the function of our enteric nervous system. With regard to interventions, I would still say that interventions during pregnancy to reduce stress, interventions in the early postnatal period, reducing stressors will definitely have long-term impacts, because we know that those periods are crucial to development of both of these systems. And while the brain, as I said, develops much longer than the ANS, these are the critical time periods that can be intervened with.
Super, great answer, thank you. There is another question. Are there any other functional disorders associated with early life stress, and how do these correspond to GI disorders? Perhaps then you might be able to handle that from Layla.
Yeah, I think what's really been studied more is psychological disorders and not so much like functional somatic syndromes, but depression, anxiety. That's really been studied, PTSD. I remember one study in fibromyalgia where they didn't feel that there was an increased early life adversity, but that was old and I'm not sure what questionnaire. But if you look at early life adversity studies, it's across many conditions, and it's not just functional conditions. It's also, COPD, CHF, diabetes, substance abuse. So this is not selected towards one specific disorder. It just shows this greater propensity for dysregulation in the system. Why somebody gets one disease versus another, I don't know. Like interstitial cystitis, they'll have more UTIs. Yeah. Fibromyalgia, they'll have a history of motor vehicle accidents. IBS is a GI infection. So there's probably some other insult on top of having that early life stress that increases the susceptibility for certain conditions.
Well, thank you. We don't have any more questions, but what I'd like to ask our attendees is, if you do think of a question after the event tonight, post it in Doc Matter and we'll ask our panelists to respond. So personally, I'd like to thank everybody for attending tonight, and also to our panelists for giving such excellent presentations. And what you're seeing on your screen now is continue the conversation from this meeting in ANMS.Matter community. There is the link to that. So with that, I guess that's all we have for tonight, and have a great evening, everyone. And thank you, our panelists, once again, for fantastic presentations. Thank you all. Goodbye.
Thank you.
Thank you. Bye-bye.